All right, so I was planning to talk about age-related changes in normal and pathological neuromagnetic signals revealed in big data. But the pathological component is maybe not ready for the prime time yet. So that will be to come. Let's see if I can move forward here. Perfect. Okay, so I just want to acknowledge that Dalhousie University is located in Mi'kma'ki, which is the ancestral and unceded territory of the Mi'kmaq people. And Halifax is Kipuktuk at the center there, shown with the arrow. And that also Nova Scotia is home to about 50 historical, often centuries old Black communities, including Africville, which is circled on the map here, whose residents were forcibly cleared in 1960. And before I get into my talk, I just wanted to share that we now have an OPM MEG system here in Halifax in my lab in this building. And so it's just some pictures from our install process, which happened during COVID, so we had to do it all ourselves, which was pretty fun. And we learned a lot of physics there. And then just showing some early data in the bottom right here. So this is a somatosensory evoked field pattern. And if you look carefully, you can see that that field is about 1 picotesla in size. So for those of you familiar with MEG data, that's a surprisingly large field, but that's the advantage of OPMs. Anyway, this is just a sidebar. I want to acknowledge funders who have supported this work before I get going, and also Lindsay Power and Brendan Brady, who are the PhD students that have done the majority of what I'm going to show, but the rest of the team as well who've helped out a lot. And Brendan is in the last, going to the last term of his PhD, so anybody who's looking for postdocs, I would highly recommend having a chat with him, and I can put you in contact with him. And I also just want to acknowledge the open science contributions to this work, M&E Python, which all of the data analysis and visualization is done through that platform. The CamCAN, which provides the data, and then GitHub, where I host most of my code. And if you're interested in the CamCAN data, you certainly can find a lot of the code to do the analysis that I'm going to show through my GitHub link. Okay, back to my title slide. I just have to move a few things around here, sorry. Okay, there we go, that's good. All right, so I just want to back up a little bit. I'm going to focus on sensory motor rhythms for this talk. And so I just want to highlight that the sensory motor cortical oscillations that we measure occur really as brief bursts of oscillatory activity. And so that's shown in that figure at the bottom left. These are intracranial recordings from a monkey, and you can see in the local field potential these oscillatory bursts that are occurring. They're about 200 milliseconds in duration. And if you look at the single unit recordings underneath, you can see that there's a fundamental shift from a stochastic firing pattern to a rhythmic firing pattern when those local field potentials are present. And so in figure B, which is the burst triggered average, so averaging on the bursts, then you can see quite clearly that the likelihood of having a spike is increased substantially during a specific component of that oscillation. So this is just sort of to show, I guess, that these rhythms that we can measure with EEG or MEG can tell us something about what's happening in the underlying cortex and the changes in the neural firing patterns there. And then the figure on the right is showing intracranial coherence between recordings at different intracranial sites in a monkey, and showing that actually these bursts synchronize across different brain areas. And so we can look at these bursts and hopefully use it as a way to probe neural network properties and learn about the mechanisms of those neural networks, and then also brain function. So this is pretty, that was in monkeys. This is some work from my own group looking at these rhythms. And what we know, and this is well studied, is that we can see mu, beta, and gamma bursts in the primary somatosensory cortices that show movement-related suppression and rebound. And so that's shown here. So this is data from an MEG channel over the primary somatosensory cortices, contralateral to a button press. And this time frequency response plot is showing time on the x-axis, frequency on the y-axis, and then the color indicates the power of the signal across many trials and many participants. And so what we can see in the baseline, so prior to the movement, at different frequencies, we have different sort of baseline amounts of signal power. And we can see that at 10 hertz, there's a lot of power, and that's because the brain generates a lot of 10 hertz activity, which we call either alpha or mu. And then we see this characteristic fall off of power as we go to higher frequencies, this 1/f effect in the power spectral density. But then as the movement occurs, we see that the power drops in the 15 to 30 hertz band. And so that's the beta suppression that we're used to talking about as a reduction in the average power during the button press. And then following the button press, there's an actual increase of the average power above the baseline values shown here. So this area is darker or redder than the baseline. And this is the so-called beta rebound. Now, if we take every frequency row and normalize to the baseline, we can re-represent this as a power change. And then some characteristics that we couldn't see very well become apparent, like this sort of orangey smudge, reddish smudge in the high frequency, which is the gamma burst that occurs at the time of the button press. But we can see quite clearly the beta suppression and then the beta rebound, and even the suppression in the 10 hertz band, which is the mu rhythm suppression, which does not have a rebound associated with it. And so we have the idea that this beta and mu suppression is a disinhibition of the motor cortex, and then a strong inhibition after the movement, which is the increased beta. And so these effects, the suppression and the rebound and the gamma burst, have been looked at in the context of aging and how those signals change with age. And so some things that have been shown for these signals is that this suppression is prolonged with increased age, that the asymmetry of mu and beta suppression, which is, so mu and beta suppression maximal contralateral to the movement, but become less asymmetric, so more equal between hemispheres with increasing age. That if we look actually at the baseline beta power, that it increases with age, and that the frequency of the beta suppression decreases with age. And so these are some effects that have been shown, but these have been shown mainly in small studies of tens of subjects, and usually in a dichotomous framework where there's young and old, or young and middle age and old participants. So we wanted to, we know now that there's lots of big data available. And so these big data repositories sort of take collections of data from a number of places, collect that data on the cloud. And so we get that data here at Dal, and then have been looking at it. In some cases, we're feeding data back into open access repositories. And so I'm going to be talking about the CamCAN data, which I think most people are familiar with, but I'm going to say a little bit more about it as we move on in the talk. So we wanted to take advantage of this big data to sort of see if the large sample size and uniform age distribution can improve our knowledge about how these rhythms are changing with age. And so we, with improved statistical power, we can get a better understanding of how these rhythms change. And maybe that can tell us a little bit more about how the brain itself is changing based on these non-invasive recordings. But then also, and I'm going to talk a lot about this, is that this framework of averaging the signal or sort of aggregating the signal over time and treating the data as though the rhythm is a sustained thing that's going all the time it oversimplifies the situation and then makes it more difficult for us to actually connect the signal that we measure to how the network itself may be changing. And so we've been looking at this transient events framework to get a better understanding of how the brain is actually changing with age. So let me talk a little bit about what I mean by a transient events framework. So this is, again, some data just that we've analyzed in the lab. And what's shown here is data from a sensor, again, over primary somatosensory cortex. And this is in a button press task. And so you can see this pattern that we expect, that there's a lot of signal in the baseline pre-movement, and then during the movement, the signal is suppressed. And then there's this rebound of the signal post-movement. And so this is, again, time on the x-axis, frequency on the y-axis, and signal power is in color. But if we look at actually the single trials, and this is just 10 trials. I think there's hundreds of trials in this average, but I'm just showing 10 trials here. We can actually see, and this is the same frequency and time scale, that there's local maxima in the time frequency response, even in single trials. And you might be able to see there's little white dots on some of those, which indicate that they are supra threshold, so they're above some threshold that we set to identify as a meaningful local maximum. And you can notice that, oh, let me go to the next slide. If I zoom in on one of them, we can sort of pick off a peak power for it. It's like the most red signal. We can look at its duration. We can look at its frequency span. And then we can also look at how many events are occurring per unit time. And so if you look sort of at these 10 trials, you'll notice that prior to time 0, there's kind of like 1 or 2 events per trial. Around time 0, there's almost none. And then after time 0, there's maybe closer to 2 per trial. And so there's actually a change in the rate of these events occurring, which is something that's hidden in the average. And so we wanted to use this framework as a way to investigate the signal, like as a trial by trial basis, or investigate the raw signal and actually extract more information that's just being lost in the average. And so this is kind of important because when we see a change in power in the average, here we have low mean power and high mean power, we can have different reasons for that change. So we can have, this is from a sort of really key paper about the transient events framework from the Jones group in the US. And so we can have an increase in mean power because we have more events happening per unit time, or we can have higher power events, the same number of events, but they have higher power. We can have that the duration of the event is expanded, or that the frequency span of the event is expanded. And in the average signal, all of these subtleties are lost. And so this might help us to understand, investigating these subtleties might help us to understand how the network's changing. And I want to give an example of that. Just give me a second. So on the right here, this is sort of a seminal paper from Birchfeller and Lopez de Silva, and they propose a circuit model for how beta rhythms are generated. And I'm not going to get into the details of it, but it's very interesting. And so what we've been doing in the past is sort of saying, okay, we had low mean power, now we have high mean power. How does this network change to lead to a change in the average spectral power? And there's actually a lot of ways that this network could change that would lead to a change in the average mean power. But now we want to ask a different question, which is, what mechanism this network would change to lead to a change in specific burst characteristics. And this is, so I'm going to just spend a little bit of time on potential different answers for these. So if we think about this circuit simply as a resonant circuit, so it outputs a burst at a specific frequency every now and then. And then if the driving signal to that circuit is happening more often, then we're going to get more of those bursts. So that would mean that the first case, event number, is increasing because the driving signal to the resonant circuit is happening more often. But if it's actually a change in event power, that's a very different thing. That means that we're getting the same amount of drive to the circuit, but that circuit just involves more neurons, maybe. It's just more neurons, and so there's more synchronous signals, and so we get a bigger signal at the end. And we can propose other explanations for event duration and frequency span. Frequency span is kind of a complicated one. What we've been thinking about that is that the underlying assumption of the analysis is that the signal is a Morley wavelet, or whatever wavelet you choose, but generally we choose a Morley wavelet to do this time frequency deconstruction. And so as the signal starts to diverge from a Morley, maybe it has some asynchrony in its dynamics. Its time frequency representation could stretch out in frequency. And so that frequency span expansion doesn't necessarily mean that the signal is including more frequencies, like it's just not as much of a Morley anymore. It looks like something else. So that leads us to also look at these signals in the time domain, and I'll get to that eventually. Anyway, so the point is that the burst characteristics allow us to make a closer connection to how the network itself is changing. If we understand the network structure, we can make that connection more closely. I just want to also mention that this is not just about the network structure and understanding the mechanics. It's also been shown that the beta rhythms actually can predict function. And so this is again study, this was both in humans and mice, showing that if you have a barely perceptible stimulus, so you perceive it like 50% of the time, that if you have decreased beta power in the baseline prior to the touch, you're more likely to perceive the stimulus. And so that's shown here in AI. The time prior to the stimulus is on the x-axis, and the mean power from an MEG source in S1 is shown on the y-axis. And misses are in red and hits are in blue. And so you can see that the signal power is higher for miss trials as compared to hit trials, which makes sense because beta is an inhibitory signal. So if you have more inhibitory signal, then you're less likely to detect a very subtle stimulus. And they show the same thing in mice. So I just thought that was really cool that they showed it across spectra, across species. What's really neat is that they also looked at burst characteristics then. So they used their transient burst framework, and they showed that this effect of higher power, meaning you're going to miss the stimulus, is actually driven by the occurrence of beta bursts. So if you have more beta bursts in the pre-stimulus interval, particularly just prior to the stimulus. So in this new figure I is time on the x-axis, probability of a burst occurring, a beta burst occurring on the y-axis. And again, red is misses. So if just before the stimulus there's a high probability that you had a beta burst, then you're more likely to miss the stimulus, as opposed to cases where you didn't have a burst right before the stimulus, then you're more likely to detect it. And so this now disentangles what is the thing about beta bursts that is causing this effect. It's just the fact that they happen in close proximity in time to the actual stimulus. We looked at, ourselves at this relationship between average power and bursts in simple button press data. And again, these are data from contralateral sensory motor areas. I showed you this plot on the left already, which is the average power change when the button press occurs. The plot on the right is actually a histogram of bursts. So we take it was the CamCAN data in this case. So we have like 600 participants, and then we find all the bursts from across the whole brain. There's like, whatever, 250,000 bursts in this plot. And then we plot the 2D histogram, so the number of events that occurred for any given time and frequency. And you can see that if you just look at them, that these plots look quite similar. You have, but some things I want to draw your attention to. Of course, this big blue blob, which is indicating that at the time of the button press, you're much less likely to have a beta burst than prior to or following. Following the button press, you have more bursts than you do prior to the button press. And so that's accounted for the rebound. And what we showed really is that you can account for most of the average power change that we see in the signal just by counting the number of bursts. Their power is changing a little bit, their durations may be changing a little bit, but the main thing is that the number of bursts that are occurring, the occurrence rate, if you will, of bursts is the real connection to the average power change. We also wanted to investigate the spatial component, spatial characteristics of these bursts. And so to do that, we developed, so this was Lindsay Power from my group who did this work. Her and I developed this localization method for individual bursts. So rather than, a lot of times, as you all probably know, when we're doing like beamform analysis, we have like an active interval and a baseline interval, and we compare those two intervals and make a map of change in power between those two intervals. But that means that if we have just rest data, we can't really make like a map because we don't have like a task on and a task off stage. So what we did was we went right into our raw time courses and we found beta bursts. So just picked a channel over contralateral primary somatosensory areas. And we did our burst detection algorithm on it. And then every time we found a burst, we said, okay, the time when the burst happens is the active interval for the beamformer. And that's like 400, just a short 400 millisecond interval around that burst. The bursts are usually around 200 milliseconds in length. And then we take the 400 milliseconds immediately prior to that as our baseline, and then we just beamform that. So we go through the whole data and we beamform all of the beta bursts that are in the data set. And we use minimum norm estimation too. And I think I'm going to show you the minimum norm estimation data actually here. So this is what it looked like. It's really interesting to look at just where these bursts are mapping to on the head. And so this was the bursts from the resting state now mapped. I think this was like for the first time that this was done. Spatial mapping of individual bursts and rest data. And you can see that the localization for left sources that were picked up from a left sensor is very much around pre and post central gyrus, as you would expect. If we do the same thing in the task data about looking at the pre-stimulus interval, it does change actually, which I can show you. So this is pre-movement bursts, and you see that the center really drops to being post-central rather than pre and post. So this is resting at rest. These bursts seem to be coming from both pre and post-central gyri, but in the pre-movement interval, it's really a post-central phenomena. And then there aren't really bursts in the movement interval, but if we look in the post-movement interval, we can see what rebound bursts look like, and those rebound bursts move to a more medial and anterior generator, which is not too surprising when you think about sort of previous literature on these things. That it would be a pre-central generator for the rebound and a post-central generator for the pre-movement activity. So all of this to say that we've been developing methods and testing out methods to understand the temporal characteristics of beta bursts and also the spatial characteristics of beta bursts. And when we had this sort of toolkit, then we said, okay, now let's take all of this into the CamCAN data and try to understand how these characteristics are changing with age. I just said that. So yeah, so here I'm going to present some of the efforts that we've been doing in the lab to answer questions about how neuroelectrophysiology changes with age using the CamCAN data and this transient burst framework. So I don't know if maybe it's a good time for any questions about this stuff so far, or should I just plow on? I know there's some stuff in the chat, but I can't see it, or I can see it. I haven't looked. Oh, just okay. Nothing so far. Okay, good. I'll push on then. So just a little bit about the data set for anyone who isn't familiar with the CamCAN data set, which is a totally reasonable thing to happen. So this is about 650 participants in this data set, which is coming from the Cambridge Centre for Aging and Neuroscience, and is shared as an open access data set. You just apply to access it, and then they give you access to whichever parts of the data you want. One of the nice characteristics of it is that it has an equal age distribution between 18 and 88. That's shown in figure A. And that distribution is consistent between male and female participants in the data set. They're predominantly right-handed, which makes sense because most people are right-handed. And there's MEG data, there's structural and functional MRI data, and then there's a whole host of behavioral data, demographic information, socioeconomic information. So there's lots of information available beyond what I'm showing. There's a host of cognitive tests data that's available per participant. And so we've been looking at the brain behavior interactions, but that's something I don't really have time. It's not really, I don't have time to talk about it. Also, it's not ready for prime time yet. But the final plot here is just showing for all the participants the mean response time in the simple button press test that they did, and nothing too interesting there. So I'm going to talk about two data sets. There's the cue button pressing task, which is the participant sees a checkerboard and or hears a pure tone cue, and then they are required to press a button with their right index finger. There's no instruction about like pressing quickly or anything. It's just like a response, a cued response with the right index finger. The interstimulus interval is varied between 2 and 26 seconds to allow for event-related fMRI analysis, which is the same paradigm was run in fMRI. They had 128 trials in total per participant, most of which were bimodal cues. And we end up using between 60 and 65 trials for our analysis because we exclude trials if the response was too late, which means they weren't really like with it on that trial too much, or because there's insufficient time between the previous trial and this trial for us to get the entire beta rebound complex, which is out to almost 2 seconds after the button press. So we need that long time. We also looked at the eyes closed wakeful resting data, which is just 7 minutes of data collected while they're eyes closed and just with no task. For simplicity, we just took the middle 3 minutes of that data because we wanted to avoid sort of edge effects of them getting into a rest or falling asleep. So we just took the middle. Sort of one of the compromises of an automated analysis of large data, I guess. So we started out with the average spectral power approach to the data and just seeing if we could before we go into transient spectral events framework, if we can just extract more information about how these signals change with age because we have more data. And so for the beta suppression, the beta rebound, and this gamma burst, we looked at the peak amplitude, so how blue or how red the signal is, the peak frequency, so which frequency bin has the peak, and then the latency of the peak in this blob for each of those blobs. And we looked at how they change with age. And based on previous literature, we had a few hypotheses. We expected a positive correlation between increasing age and the following signals: decreasing beta rebound amplitude, so this signal getting smaller, decreasing beta rebound frequency, so that the peak frequency going down is sort of a cortical slowing, if you will, and then decreasing beta suppression frequency, so this one also going down. I'm going to maybe skip the details of the analysis for time and just get to the results. So what we found is for beta suppression, the localization was left hemisphere pre and post central sulcus gyrus, and we found that the beta suppression magnitude increased with age, that the rebound amplitude decreased with age, and that the rebound frequency decreased with age. And so two of these were effects we hypothesized. We didn't see that the beta suppression frequency decreased with age, so we didn't see that effect here in B. And an effect that we didn't expect was that the beta suppression amplitude would increase. And so this increasing amplitude of the beta suppression and decreasing rebound made us think that probably the pre-movement beta levels were increasing. But we wanted to test that, and so we looked at that in this beta burst framework. So I guess what we wanted to do with the follow-up studies was then to link the beta bursts to what we found. So how did transient beta events modulate suppression and rebound that we measured in amplitude? How did the beta events modulate suppression and rebound frequency? So is this like that the peak frequency shifts, or that there's more low frequency bursts? It could be either one, so we want to look into that. And then what can those changes in burst characteristics tell us about the underlying network? So we took the CamCAN data, we detected bursts over S1 and M1 during the motor task and the rest task. We did some source estimation of those beta bursts, and then identified age-related changes in the spatial and temporal characteristics. And we expected that the age-related trends in the transient burst temporal characteristics would reflect what we've already seen in the average power change. And we also expected that older participants would show a more widespread activation than younger participants. And this is kind of based on some fMRI work that we expected that. And so we took this transient burst framework that I've described and then characterized the roughly quarter million bursts that are in the data set, and then looked at age-related changes in those. And so this quarter million bursts really changes the game in terms of like our ability to do statistical analysis. So here's what we found for the task data in terms of how beta bursts are changing. And so there's a lot here. The first column is the pre-movement interval, how that signals or characteristics are changing there. The middle column is the movement interval, and then the final right one is post-movement, so the rebound interval. And then the top row is the burst rate, so the number of bursts per second. Normalized peak power, so that's how strong are the bursts. And the peak frequency of the bursts, and then the frequency span, which is a complicated signal, again, to understand. So we saw that the burst rate increases prior to movement, but then decreases following movement. We saw that there were quadratic trends, which we hadn't looked at before. And in a dichotomized study, you wouldn't be able to look at these. So this is a really new finding. We found that the peak power decreases both pre-movement and post-movement, and that frequency span shows a complicated behavior, but it's sort of lowest in middle age. When we looked at the localization of these bursts, what's shown here is, so for the post-movement interval, we did this for every interval, but what we did was we took each individual's data burst map, and then we found the peak location for where their bursts are coming from. And then we plotted that on a template brain. And so in this plot, the color of each dot represents the peak location of a beta bursts for an individual. And the color is age. So what you can see is that as you go from posterior to anterior, you also go from blue to red, meaning that older participants are more likely to have an anterior beta burst peak location. And that's also shown on the right, where we just show the posterior position as a function of age. So just another representation of the same thing. So it seems like the generator actually of these bursts is even changing in where it is in the brain with age. And then if we look at the whole map of the beta burst rather than just the peak location, what we see is that frontal areas get involved, or frontal areas are showing a change with age. And so we see, so what's shown here is different ROIs, and the color is indicating the T statistic of a test to see if there's a relationship between the activity in the ROI and age. And so the more yellow it is, the stronger the correlation between age and activity in that region of interest. So it's really frontal areas that are showing a change. And that for some of those areas I've shown on the right here, age on the x-axis and then the mean activation in the ROI. And so you can see that there's clearly quadratic effects here where as you age, frontal areas get involved, and then that just sort of, I guess, plateaus out to older ages. So we know that the peak location is changing, but also that the activation is spreading out more. And the interpretation of that is complex in MEG data. So there's a lot of reasons why signal can spread, but that's something to investigate further. In the resting state, we see a different set of changes. So we see that the burst rate is increasing with age. So this is age on the x-axis and burst rate on the y-axis. We could see a substantial increase in burst rate from youth into middle age that tops out, plateaus around 60. And actually, a lot of the effects that I'm showing that are quadratic have peaks in and around 60. We developed a way to do burst triggered averaging like people do in invasive recordings for our MEG data. And so this is my student Brendan, who developed a sort of labeling of the beta burst components as beta not, beta 1, beta 2, beta 3, beta 4, going positive in time and then negative in time. So all the bursts are averaged on this central beta not peak. And we looked at how the characteristics of these peaks changes with age to get away from this Morley assumption. And so what he found is that the peak times of these peaks sort of spreads out with age. So here age is on the x-axis again, and peak times on the y-axis. And so for the plus peaks, that's going more positive. And for the minus peaks, it's going more negative. So that this wave is getting sort of spreading out in time, which would be a slowing, like it would come out as a reduction in the peak frequency. But also that the amplitudes of some of the peaks are increasing. So beta +1 and beta -1 are increasing in magnitude with increasing age. So we're seeing that the beta bursts sort of temporal dynamics, if you will, are changing with age. So this is lots of information to help us sort of start to make that link to the network that I showed before. And this is just sort of a summary slide of everything that Brendan showed, but he looked at actually 4 regions of interest, and is showing sort of how the signals change with age. So the durations aren't really changing, but the signal is sort of spreading out in time. So it's spreading out in time, which means there's less cycles actually, because it's the same duration. As the frequency drops, that means there's less cycles in that pulse. And they're happening more regularly. The pulses are happening more regularly with increasing age. So this was just sort of a summary visualization. And this is just to show that this movement of the peak forward, which I showed for bursts that happen in the rebound, also happens in the resting state, but it's a little less a focal result. And actually, I think it's hard to see, so sorry about that. But we see, again, sort of movement of the peak burst location towards the precentral gyrus as you age, or for older participants, I should say. So, yeah, so there's lots of things happening here. The burst rate is increasing at rest. It's increasing prior to movement, but it's decreasing following movement. The peak power is highest in middle age. The peak frequency is decreasing. We're seeing that the frequency span is lowest in middle age, and maybe that's because the burst triggered average actually looks different. It doesn't look like a Morley anymore. It's got less cycles in it. We're seeing an anterior shift of the peak activation with age, and we're also seeing the recruitment of frontal activity into middle age. So this is now a whole host of behaviors that we can start to link to, and I haven't done this yet, to link to this network schematic. This is like 1999, which is doesn't seem like a long time ago to me, but it does to some people. There's a newer network model from the Jones group again, which is a computational network model. So you can actually, this is a more complex model with like many neurons in it that are actually interacting on the computer. And you can actually play with the dials of this of the components of this network, like the proximal drive and the distal drive and the all kinds of stuff, all kinds of things, and generate time courses of mu and beta activity. So we can actually get into tuning these mechanisms to see if we can generate behavior like I have listed here. And that might help us to test hypotheses about how the network is changing. Admittedly in a computational space, but it's an interesting direction to move forward. So that brings me to the end. So just to summarize, it seems like analyzing the characteristics of transient events in the raw data opens the door to clearer links to the underlying mechanisms. But we need certain expertise to try to understand these links. I'm a physicist. I got into brain imaging by accident. I need people who know this stuff really well to help me to make the connections between our experimental findings and how neural networks work at a very low level. It's pretty clear that big data has provided improved statistical power for investigating age-related changes when we compare what we're seeing to these sort of small sample size dichotomous studies that were what we had a lot before we had open access data. The burst changes that we've seen are listed here again, and I'm not going to go through them. So what are we thinking about next? So there's, like I said, I want to link these characteristics to underlying network mechanisms. There's also lots of other bursts beside for beta. And so there's lots of, now that we have this framework, we can look at alpha and mu and gamma and all that stuff. So, and we're also looking at, Brendan just published an analysis of the aperiodic components of the data, which I didn't have time to talk about, but also show age-related changes. We want to combine the spatial and temporal changes into one framework. So right now we sort of like look at a time course for temporal changes, and then we look at the whole map for spatial changes, but I want to bring those together a multivariate framework. So that's something that we're actively working on right now. And then, like I said, there's lots of other data in the CamCAN data set. So we're starting to look at things like cognitive performance and how that relates to different transients in the data. And so hopefully I can talk about that next time. It's kind of preliminary and I didn't want to get into it at this stage, but particularly because this might go on YouTube. So I don't know if there's anything wrong or untested. Yeah, so that's where we are and where we're thinking about going. So again, just thanks to everyone who's been involved in this work, and glad to take any questions.